Please turn on your cameras and let's begin today's lesson. Today's lesson is going to be a little bit difficult, the second part of it, because we're going to talk about blood vessels, formations of the head and neck and other blood vessels. But let's get rid of this easy two topics, development of the skull and vertebral column. Okay, you pretty much know that skull and vertebral column, they belong to the axial skeleton, right? So they're part of the axial skeleton. We have learned it already, but axial skeleton will arise from the paraxial mesoderm. Okay, it will come from the paraxial mesoderm. Maybe you remember that we always draw it like this. Whenever you draw a 3-week-old embryo, you will always draw ectoderm at the top, right? So right under it, there is a notochord. Right under it, there is an endoderm. And do not forget which cavity is at the top. Which cavity do we have here?
Amniotic cavity.
Amniotic cavity is going to be here. In embryo 1, you have learned that amniotic cavity is lined by the amnioblasts, and these amnioblasts have arisen from epiblast when it was a bilaminar germ disc, right? So when your embryo was a bilaminar germ disc. Endoderm, which is the third layer right here, is not only present in the trilaminar germ disc, but you also remember that it is part of the wall of the yolk sac as well, right? So here within the wall of the yolk sac, we are also going to have the endoderm as well. What is important for us right now is what is on the two sides of the notochord. So right on the two sides of it, we have the paraxial mesoderm. And in embryo 1, you should have learned that paraxial mesoderm is organized into somites. Okay, that it has this particular arrangement that are called somites. Each somite, and there are many of them, okay, so we start making somites approximately from day 20, and we finish making them approximately around 5th week. And by the 5th week, you have made around 41 to 42 somites. And this number is a bit too high. So some somites are going to degenerate. They are going to disappear. Which ones disappear? The first occipital somite, okay, so the ones which are in your occipital region, the two somites here, they are going to degenerate, disappear. And also 5 to 7 coccygeal somites will also disappear. If you do the math, right, so if you see how many we have removed and how many have left, these somites, the number of these somites, correspond to the number of your vertebrae. So I want you to remember that whenever you hear the word somite, think vertebra. Okay, so think of them as the future vertebrae. Now, what is on the two sides of the somite? In the cephalic region, we do not have this tissue. So somewhere from the future thoracic region, that's this thing will appear. This is the intermediate mesoderm is going to be present next to the paraxial mesoderm. Okay, so it's going to be again from the thoracic region. In the head, we do not have intermediate mesoderm, because you remember that intermediate mesoderm will become urinary and genital system. So of course, in the cephalic region, in your head, it's impossible for the urinary system to develop, so it cannot be present in the cephalic region. And lastly, on the two sides of the intermediate mesoderm, we have a V-shaped structure, which is known as the we talked about it twice already. Every lesson we talk about it a little bit.
Lateral plate.
Yes, this is a lateral plate mesoderm. Could you tell me how do we call this one of the lateral plate?
Parietal.
Parietal mesoderm, and this one will be
Visceral.
visceral. This is ectoderm right here, right? So how do we call parietal mesoderm together with the ectoderm? How do we call it?
Somatopleural?
Yes, it's called somatopleur. And visceral mesoderm of the lateral plate together with the underlying endoderm is going to be splanchnopleur, right? So this is important. I told you it's quite important. We will need it in the rest of the embryo too. Because we're talking about development of the skull and vertebral column, of course, the most important part for us is going to be the paraxial mesoderm itself. I strongly recommend you guys to really refresh your embryo one in terms of how exactly did we make the somites, okay? So how did they appear? Because they are segmented, right? I repeat, we made 41 to 42 of them, but then some of them disappeared. So this is an embryo one knowledge, really to refresh that. What does each somite of the paraxial mesoderm consist of? This picture is a very good one because it really does represent everything that we see in the somite. So this right here, this is going to be your one somite of the paraxial mesoderm. How many are there? Well, I repeat, on 5th week, we had 41 to 42 pairs, right? Because notochord between them. So 41 to 42 pairs of somites were present. What does each somite consist of? Some cells are responsible for creating dermis of the skin. Last lesson we discussed it a little bit, right? Whenever you say that ectoderm, the blue line, whenever you say that ectoderm will give rise to skin, you should be careful because ectoderm only gives rise to the epidermis. Only the top layer of the skin will come from the ectoderm. Dermis will come from the dermatome. And in the anatomy, you also have this term, right? You have a term in the anatomy called dermatome. What is dermatome in the anatomy? It is part of the skin which is innervated by a particular cutaneous nerve. In your atlas, it almost looks like a zebra appearance, right? So the entire human body has a zebra appearance because each of the cutaneous nerves innervates only a particular part of the skin. So this is what a dermatome in the anatomy is. Dermatome in the embryology, it's a part of the somite which is responsible for giving rise to the dermis of that particular level. So for example, if I'm talking about C3 somite, right? I'm talking about C3 somite, the C3 dermatome will give rise to the dermis of this region specifically, right? So I think it's quite clear. Under the dermatome, the bigger portion of the somite is sclerotome. And sclerotome is the one which is important for making skull and vertebral column. Okay, so this is the one which is important. Sclerotome consists of cells which will differentiate to become chondroblasts or osteoblasts, depending which kind of bone are we talking about. Okay, so we'll talk about it right now. So sclerotomes here, we have some kind of stem cells here, and they can become two things, either a chondroblast or an osteoblast. Of course, you remember that chondroblasts make cartilage. If I make cartilage first, which kind of ossification am I talking about?
Endochondral.
Endochondral. If I'm making osteoblast first, right? So this is going to be the other one, which is the intramembranous ossification. So sclerotome is important for us. Somewhere on the sides between the dermatome and the sclerotome, there are myotomes. And these myotomes, of course, are going to be responsible for giving rise to the muscle of the axial skeleton. So this picture is important. Really try to take a sort of mental picture of it so it stays in your brain. You have the overlying ectoderm, you have a neural tube which is your future spinal cord, right? And right under it, there is a notochord. How do we call the remnant of the notochord?
Nucleus pulposus.
Nucleus pulposus of the intervertebral disc, right? So do not forget that it's going to be the nucleus pulposus of the intervertebral disc. Okay, sorry guys. Yeah, so nucleus pulposus of the intervertebral disc, because intervertebral disc is going to have two parts. The center, which is a jelly, the hyaluronic jelly called nucleus pulposus, and it has the periphery, which is a fibrocartilage, the annulus fibrosus, right? So that's the two parts of the intervertebral disc. So notochord will only give rise, or will only remain as the nucleus pulposus. This is a friendly reminder to really open the histo book, open the histo 1 notes if you have any, and really try to remember the intramembranous and the endochondral ossifications. The problem with the embryo topics that majority of them will lead you here, majority of them, okay? So nothing stops them from asking you, could you tell me the steps of endochondral ossification? Could you tell me the steps of intramembranous ossification? If I have to be honest with you, usually that's the failing question, okay? Because they know that you didn't revise histo 1. So if they just wanna, get rid of you, like you haven't been doing good in the exam, so they just wanna get rid of you quickly, they'll ask you this question. So really, please refresh your histo 1 for this, okay? So for endochondral or intramembranous ossification. But sort of to help you, I think I told you this actually before in our histo lessons, if you took it with me, intramembranous ossification is for the bones that will not bear any weight. So if they cannot bear any weight, so for example, your face, right? Or your calvaria, these ones. So they don't bear any weight. So they must arise via the intramembranous ossification. Endochondral ossification is for the bones that do bear weight. So this is your long bones, your vertebral column, pelvis, right? So all of this kind of stuff. The base of the skull, so yeah. So generally speaking, skull can be divided into two parts. We have the neurocranium and we have the viscerocranium. Viscerocranium, this is the skeleton of the face, and we have covered it yesterday when we did the pharyngeal arches. So if they ask you about the viscerocranium, how did some of the bones on your face appear, all you have to do is talk about pharyngeal arches, and do not forget that your frontal bone, which is right here, will be the product of the frontonasal prominence, right? We talked about it yesterday. There was a huge prominence here with a bunch of neural crest cells present there. So yeah, so viscerocranium, pharyngeal arches. Neurocranium, this is the protective case around your brain, and it will also consist of two parts. There is the membranous part and there is the cartilaginous part. Of course, from the two names, you can understand what's the difference between them. Membranous part, these are the flat bones, the calvaria that we have around your brain, and these bones arise via intramembranous ossification because they do not bear any weight whatsoever. Cartilaginous part, which is also known as the chondrocranium, right, of the neurocranium, is the base of the skull. So that part of the skull on which brain sits, right? So the base of the skull. And it arises via the endochondral ossification. If they ask you why, right? So why neurocranium has two parts, the membranous and the cartilaginous one? Well, brain is heavy, right? So brain is quite heavy. So the base of the skull has to arise via endochondral ossification because it does bear weight. The weight that it bears is the weight of the brain, which is sitting on it. So that's the major difference between them. So how did we make membranous neurocranium, right? So how did we make the flat bones? two sources, neural crest cells and paraxial mesoderm. I don't think I mentioned it, but we have two things here. We have somatomeres and we have somites, and they are actually a bit different. Somites are from the occipital region downwards, and they consist of these things, of dermatome, myotome, sclerotome. Okay, so somites, they have it. Somatomeres do not have it. They are just in the head region, present here like that, and it is just paraxial mesoderm. It doesn't really contain the dermatome, myotome, or sclerotome. So it's not like, it doesn't have this particular division compared to somites. Okay, so again, to simplify, somites are these, right? So they have the dermatome, myotome, sclerotome. Somatomeres are in the head region, and they're just clusters of like paraxial mesoderm that is not separated into these particular segments. So in the head region, we're going to have these paraxial mesoderm somatomeres together with the neural crest cells. I told you this in the first or second lesson, that if you look at the body as a whole, always neural crest cells love to accumulate cranially, cephalically, in your head region. That's where most of the neural crest cells are going to be. You will not find them in the pelvic region. Okay, you will not find them there. Majority of them are sitting in the head region. So mesenchyme from these two sources, so these two things will convert to a mesenchyme, embryonic connective tissue. And this mesenchyme will start to create some kind of centers. And the name of these centers depend on the name of the future bone. So for example, if I have a mesenchymal center in this region, you will call this mesenchymal region as the parietal mesenchymal region, right? So if it is here, occipital, which is there, frontal, it doesn't really have a specific name, just mesenchymal centers. Because it is intramembranous ossification, what's going to happen is that these mesenchymal stem cells will immediately convert to osteoblasts. These osteoblasts start to secrete osteoid in the radiating manner, right? So they're going to be spreading this osteoid towards the periphery until this osteoid will meet up with another center's osteoid, right? So the part where they will meet up. How do you call, like what is it called? The meeting between two osteoid centers. How do you call this?
Suture.
Suture. And how do you call the meeting point between more than two osteoid centers? So between two specifically, it is a suture, but between more than two, it's called, we have one here, well, we had one here, we had one here, and we had one here. Fontanelle. Okay, so they are called fontanelle. It's actually a very important question in the anatomy. They will ask you, what is the difference between fontanelle and a suture? Suture is the meeting point between two bones. If it is more than two bones that are meeting in that region, this is known as the fontanelle. So that's it. That's how you make your calvaria. Okay, so I think it's very easy. Those bones, spicules, spacules, whatever, it's just the radiating osteoid. Remember from histo 1 that osteoid is unmineralized bone, right? So it will mineralize later because the head will be expanding thanks to the brain, right? The growth of the brain. So you don't really make the bone immediately. You first make the osteoid and then later, much later, it will ossify. Cartilaginous neurocranium is the base of the skull, and it is kind of divided into two. And the point of the division is going to be the sella turcica. Okay, you can see that sella turcica or pituitary fossa is the point where the two sources are meeting up. In blue, this is neural crest cells. In orange, this is paraxial mesoderm. So the same idea, just that here it was sort of a mix, right? So they were everywhere. It was a mix between paraxial mesoderm and neural crest cell. There was no division between them. In the cartilaginous part, in the base of the skull, there is a division. So majority of the neural crest cells will be more cephalically, right? So, or more anteriorly, if you must say so, and paraxial mesoderm will be more posteriorly. What's going to happen? The two sources will become mesenchyme. Mesenchyme will become chondroblast. Chondroblast will make a cartilage in the shape of these structures. Luckily, we don't have to know why exactly. And as the brain will be developing, it will modify this cartilage to the most convenient way. So for example, if a certain, I don't know, nerve has to go in a certain direction, luckily it's a cartilage, so it will just push through it, right, and will reach the destination. Think of it as this mold, okay? So this cartilage that you have here at the base of the skull, it's quite soft, so you can do things to it. The brain itself will modify the way the base of the skull will look like later, right? So it will become the bone itself. So some new osteoblasts will appear there. They will secrete osteoid, add calcium, and it will become bone. Okay, fontanelles. Okay, we have anterior fontanelle, posterior fontanelle. We have the anterolateral or sphenoidal, or posterolateral or mastoid. Okay, so these are the four fontanelles which are present on the skull. These numbers are important. So they're going to ask you when are some of the fontanelles are going to be closing. Anterior fontanelle will be closing by 1.5 years, right? Posterior fontanelle 1 to 2 months of age. It's of course after birth, right? Sphenoidal fontanelle 6 months after birth, right? And mastoid 6 to 18 months after birth. Fun fact, anatomy or embryology is a theoretical subject. So these numbers are false. Okay, so they are wrong. When you're going to come in the 4th year to obstetrics and gynecology, majority of the fontanelles have fused except for the anterior. So by the time you're born, majority of them have closed up. Okay, all 3 of them, the posterior, anterolateral, and posterolateral, have closed up. They have fused, that's it, except for the anterior fontanelle. You can actually see it here. Okay, so this is the anterior fontanelle. If you touch it, you're touching the brain, right? So there's a brain behind it. There's just a little bit of the fibrocartilage present here, but it will be ossified in the later stages. So I'm trying to tell you that for the 4th year, remember that these numbers are false, maybe except for the anterior fontanelle, but anterior fontanelle typically closes by the 1 year of age, like even 10 months of age, okay, not 1.5 years. What is the purpose of anterior, what is the purpose of fontanelles? They are allowing the overlap of the front of the flat bones during birth. So the circumference of the head is much bigger than the circumference of the vagina. Okay, so it's much bigger. So vagina will maximum dilate to 10 centimeters, right? So that's when we tell the mother to push, maximum 10 centimeters. The head, when you're born, is usually about 12 to 15 centimeters. So how do you push 12 to 15 out of the 10? You can't. So that's why it really does hurt a lot, right? So sometimes they do the episiotomy where they cut the vagina towards your anal region, which, fun fact, repairs itself within 2 days. In 2 days, it's fully repaired. It's like one of the fastest repair of the body. So to be able to push 15 centimeters out of the 10 centimeters, you have to allow the bones to do this. Okay, so if you have ever seen the head of the newborn, it doesn't look like ours. It's almost like an egg shape. Okay, it's very elongated head, like literally like egg people. I don't know if you're young enough or old enough, but back in when I was a teenager, we had this egg people movie, very like it was 80s or 90s old movie. So it was exactly how a newborn would look like, very elongated head like this. Okay, of course it comes back to normal. Of course. So you don't stay like that. It will come to normal. The swelling will come down. So it will be fine. So the purpose of the fontanelles is to allow the bones to overlap so you can have a normal vaginal delivery. This is a very weird question, but sometimes they ask. The two huge sutures that we have, the sagittal and the coronal sutures, they have a different origin. Okay, so at first, of course, you know that it's a cartilage at first, and then that cartilage will ossify with fibrocartilage. So sagittal suture is going to be arising from the neural crest cells, and the paraxial mesoderm will give rise to the coronal suture. So remember that. Now, vertebrae and vertebral column. Okay, I need to draw it to be able to show you. Okay, so everything that I will talk about is written right here. So let's just try to draw it because that's the only way to really describe what is happening there. How is the vertebral column formed, right? So how are the vertebrae are going to be formed? We have a neural tube. As you remember, neural tube will become the spinal cord. Anterior to the neural tube, we have a notochord, right? So this is a notochord right here. It's a very different positioning because usually they do something like this, right? Which is a cross section, so neural tube and notochord, but really try to understand, right? So this is a neural tube and this is the notochord. On the two sides of the neural tube and notochord, we have the paraxial mesoderm somites, right? So they're usually paired structures, and very important is that they are segmented. So there are different segments of them. I repeat, fifth week, we have made 41 to 42 segments, but it's too many. We will remove some, right? So let's draw, I don't know, 3 somites that are 3 pairs of somites, right? So how do we make a vertebra? We don't need the entire somite because remember that each of your somite will have a myotome, dermatome, and a sclerotome. To make a vertebra, we only need the sclerotome part. So sclerotomes of the 2 somites that are at the same level, the sclerotome cells are starting to migrate around the neural tube and the notochord and fuse. Okay, so there is a one particular fused sclerotome portion, and then there is another one a little bit lower, right? So sclerotome cells are migrating around the neural tube and the notochord and fusing together. So if we have to redraw what has happened, we still have your neural tube. We still have the notochord that is here, but now we have fused segments, fused sclerotomal segments, right? So there's going to be, I don't know, there's going to be one that is here like this, right? There's going to be another that is here, and there's going to be another. So like thick bricks of tissue. When you look at it, it might look like you have made your vertebrae, right? Because they kind of look like your future vertebrae. So they're very much separated, and there's like some gaps between them. Of course, we have discussed this right now. What are the sclerotome cells are going to do? They're going to turn to become chondroblasts. These chondroblasts are going to make a cartilage. This cartilage is going to be in the shape of the vertebrae. And then as your embryo is growing, the shape will be modified depending on the position of the embryo, right? So embryo will modify its appearance, the vertebrae appearance. So these fused segments that we have here now have to be resegmented. And I truly wish that all of you get this topic, okay? So development of the vertebral column, because all you have to say, it's just one word, resegmentation. Development of the vertebral column, resegmentation. So these fused sclerotomal segments have to be resegmented. They have to be broken down and put together again. So what am I talking about? Here we have a head somewhere in this region, and here we have a tail. These are your fused sclerotomal segments. And for now we have 3, for example. Resegmentation begins when each of these segments starts to separate into 2. They start to kind of divide into 2. The top one, we're going to call it the cephalic, and the bottom one is the caudal, right? So you know that cephalic means closer to the head and caudal means closer to the tail. So each of this sclerotomal fused segment divides into 2. The upper one is the cephalic, the lower one is the caudal. So what is resegmentation? You can guess immediately which ones are going to now fuse. Of course, these 2 will fuse together, and then these 2 will fuse together. So basically, resegmentation is when caudal half, right? So caudal half of the upper segment fuses with the cephalic half of the lower segment. So now I have a newly formed segment here. Again, resegmentation. Caudal half of the upper segment fuses with the lower segment's cephalic half. So now I have another segment. So it's repositioned. Because it is repositioned, now the arteries and the nerves are entering at an angle, at an angle like this. Okay, so now they're entering at this particular angle. Before they were straight, now because of the resegmentation, it has reshaped, and now there's like almost like a, I don't know, 45, 35 degree angle. To prevent further resegmentation, to make sure that it doesn't continue, mesenchyme is going to come in and occupy these gaps. This mesenchyme will, of course, come from the paraxial mesoderm. Okay, so like some leftover paraxial cells, we're gonna give rise to some of this mesenchyme. And this mesenchyme that is coming in and occupying this gap region will be responsible for giving rise to the annulus fibrosus of the intervertebral disc. Do not forget that in the center of this structure, we have a notochord, right? Because if you look at this picture, notochord is always sort of in the center. Neural tube is a bit behind, it's a bit more posterior. So in the center of this structure, there is a notochord, and this notochord will degenerate everywhere except in the centers of your future intervertebral discs. That's where it will stay. It will stay there as the nucleus pulposus of the intervertebral disc. Okay, one more thing, Hox genes. You have to know that this entire process, the different shapes of the vertebra, different, I don't know, everything of the vertebra is regulated by Hox genes. When do we start making the vertebral column? Very easy, 4th week. I told you in the previous lesson that whenever you are unsure which organ develops when, which structure develops when, just say 4th week and there's an 80% chance that you're right. Okay, so 4th week. two curvatures are present, two types of curvatures that are present. We have the primary and the secondary curvatures. Primary curvatures are the ones that you are born with, right? While secondary are the acquired curvatures. The primary curvatures that we have are the thoracic and the sacral curvatures. You're already born with them. You made them as an embryo. Secondary curvatures, the first one to appear is the cervical curvature. It appears when the baby learns to hold the head. Okay, so this usually happens, well, nowadays babies are very, very active, so they learn to hold the head much earlier, but by the book it should happen about 2 to 3 months of age. Okay, so this is when the baby spontaneously learns to turn around on baby's belly and learns to hold the head up like this. Okay, so this is when the cervical curvature will appear. Lumbar curvature appears when the baby learns how to walk, and as we know that girls usually walk sooner than boys. Average age for girls to walk is about 11 to 12 months. For boys is about 13 to 14 months. Okay, so again, it's very subjective. It's just some average numbers for you. So lumbar curvature appears when you learn how to walk. So that's why when the baby learns to walk, it's very important that the baby learns how to walk right, meaning that they're not just tiptoeing, okay, but you have to make sure that the baby puts the entire foot on the ground because it may affect the curvature. Okay. Okay, we're done with this part. Just 2 minutes break. I'm gonna quickly use the bathroom and we will continue. Okay, so let's continue. Development of the blood vessels. Back in the day, this used to be one topic. It was topic number one. Luckily, they have broken it down into like almost 6 or 7. Okay, so please remind me today to, actually, I'm going to do it immediately after today's lesson. I'm going to highlight the topics that we have done so far. Okay, so you can try to find them because yesterday I forgot, I'm sorry. So luckily, they have broken down this particular huge topic into several. So let's take a look. We have kind of discussed this already, that generally speaking, mesoderm is the one which is very important for making a blood vessel and making blood cells. I told you in the revision of the embryo 1 that whenever you think of mesoderm, right? So think about it this way. We know that paraxial mesoderm gives rise to the axial skeleton, like your skull and vertebral column, yes. But also remember that because it is a mesoderm, it also will give rise to all of the connective tissue there, right? And all of the blood vessels and blood. So always think about it this way. Mesoderm will give rise to something big in that particular region, like paraxial mesoderm, but it will always be giving rise to blood and the connective tissue in that region as well. There are two possible ways for a blood vessel to arise. One of them is called vascular genesis, and one of them is called angiogenesis. Vascular genesis is only present in embryo's life. So as you're an embryo, you will have vascular genesis. Angiogenesis could be present in embryo's life, but also in you. I don't want to lie, but as far as I remember, for every kilogram of fat that you gain, of weight, right, of fat that you gain, there are additional 5 or 10 kilometers of the blood vessels that you will never get rid of. That's why you can lose weight, but the body remembers that weight. So it can immediately gain it back if you don't control yourself because of its blood vessels, because they will not go anywhere, they will stay there. Okay, so this is why getting rid of fat is very, very difficult. So angiogenesis is not only fetal process, it's also an adult process as well. Okay, so we also have it not only for fat gaining, but you know, our lifestyle might be very bad. We may eat very bad, smoke, I don't know, alcohol. One little blood vessel may get clogged, so another one will appear. So it's a very constant, continuous kind of process. So let's try to understand how does it happen exactly. So in embryo, actually we start making blood as early as day 14. Maybe you remember from our embryo 1 revision, when we made the secondary yolk sac, the outside of the secondary yolk sac was extra embryonic splanchnopleuric mesoderm, extra embryonic splanchnopleuric mesoderm. And that mesoderm was the first one to start making blood. And what's interesting is this: do you make a blood vessel first or blood? The answer is blood. You first make blood. So you first make red blood cells, right? You first make platelets, plasma, and only then you will put this blood into a vessel. So it's like the never-ending discussion: what comes first, egg or chicken? Here, it's the analogy is what comes first, blood vessel or blood? But we actually have an answer. Okay, so blood actually comes first. So approximately starting from day 14, little tiny blood islands start to accumulate. Okay, so just random places, they're accumulating in random places, some blood islands, little tiny islands. Then certain cells come in, which are called angioblasts. Angioblasts arise from the primordial mesenchymal cells. Where does the mesenchyme come from? Well, this is embryonic connective tissue, and we know that connective tissue comes from mesoderm, right? It comes from mesoderm. Primordial mesenchymal cells do not come from mesoderm, they come from epiblast. They're one of the first ones, the great-great-grandmother of the mesenchymal tissue, okay? So they come from the epiblast. So these primordial mesenchymal cells, aka angioblasts, come in and they literally start to line these blood islands. Okay, they start to sort of put them together into some kind of vessel of some sort, right? So these cells that I'm drawing for you, these are the angioblasts. And they just come in and they sort of put these blood islands into one place, into a primitive vessel. So these are your angioblasts. The question is, why angioblasts, for example, go in this direction and not in that direction, right? So why are they forming a vessel in a very particular direction? The answer is this growth factor, which is known as the vascular endothelial growth factor. This is a growth factor which is responsible for patterning your entire vascular system. So whichever way it's present, that way the blood vessel will go. Okay, so I don't want to say it's random because it's not random. It is very highly regulated. Just we do not learn the regulation. Okay, so why exactly the VEGF will appear that way? Just know that because of the vascular endothelial growth factor, the blood vessel will go in a certain direction. It's like, you know, when the airplane is landing, yeah, there are lights that are helping the airplane to go in a certain direction. So that those lights are your VEGF. They're literally just telling the blood vessels, specifically the angioblast, which way should they go. Okay, so that's the VEGF. Fun fact about the vascular endothelial growth factor, that cancer cells secrete it, and that's how they spread, you know, metastasis, right? So the spread of the cancer, there could be two possible ways, the lymphatic or the vascular. The vascular is thanks to this one, thanks to the VEGF. The cancer cells secrete their own VEGF, make a vessel connecting to a patient's blood vessel, and then detach, spread, and infect the rest of the body. So they're very interesting therapy right now that inhibits this one, and it's actually helping with the cancer, that if you inhibit the VEGF, it doesn't really spread as much. So what about angiogenesis? So what we have discussed right now, this is a vascular genesis. This is making a big vessel. So the big vessels in embryo, these are, for example, cardinal veins and dorsal aorta, for example. Okay, so cardinal veins, dorsal aorta. I know these vessels mean nothing to you right now, but we will learn them right now. Okay, so just huge vessels in embryo. That's how they formed, the vascular genesis. What about angiogenesis? Well, imagine that like there is a little tiny branch from this vessel, right? And then there's another branch from that one. So that's your angiogenesis. So the remainder of the vascular system, like a tree that is branching from one particular trunk, that's your angiogenesis. So difference between them, huge vessels are made by vascular genesis, right? So like your aorta is made by vascular genesis, like your, I don't know, inferior vena cava, probably vascular genesis is as well, right? The rest of the little vessels will be the angiogenesis. Of course, embryo will also have arterial and venous system. Let's talk about arterial system first. Arterial system of the embryo consists of three types of vessels. So that's what I wrote for you here. We have aortic arches, we have vitelline and umbilical arteries, and we have coronary arteries. So let's talk about aortic arches first. To understand it, we need to discuss how does the heart of the embryo looks like when the embryo is approximately 4 weeks old. Okay, so how does the heart of the embryo looks like? Embryo is 4 weeks old. Heart looks like a tube. Okay, it has a tube-like appearance. So not your normal 4-chamber structure, like a tube. This heart tube has 2 ends. The cephalic end, right, so the top, is the arterial end. Okay, so this is the arterial end. And the bottom, the caudal end of the heart tube, this is the venous end. So let's indicate, right? So this is the venous end of the heart tube. This is the arterial end of the heart tube. The venous end of the heart tube has 2 branches, 2 bigger branches. We call those 2 branches as sinus horns, like a bull, you know, sinus horns. So this is, for example, a right sinus horn, and this is left sinus horn. This entire end of the heart tube, so all of this, right, so with the sinus horns, this is known as the sinus venosum. Okay, so this is known as the sinus venosum of the heart tube. So could you tell me which bigger vessel that you know will be emptying here into these horns. And these vessels come from placenta. So the direction is like this way. They're coming from placenta into the sinus venosum. Which vessels? Come on, histo.
Umbilical arteries.
Umbilical
Vein.
vein. Because as you know, in embryo, it is a little bit the opposite, right?
Yeah.
The umbilical arteries technically carry deoxygenated blood, right? So they technically carry the venous blood. And umbilical veins technically carry the arterial. So when I say that this is venous end of the heart tube, do I really mean that it is the venous end of the heart tube? Because the blood that is entering here is anything but venous. Okay, it's umbilical veins. They're coming from placenta. So the blood that is entering into the sinus venosum is actually quite good blood. It's a very good blood. The problem is that there is another vein entering here, and this is going to be the vitelline vein. And vitelline vein, you're gonna see in the today or in the next lesson, is carrying venous blood. So it's actually carrying venous blood, not arterial, but venous blood. So sinus venosum of the heart tube is the first place where mixing of blood occurs, where arterial blood, which is coming from the umbilical veins from the placenta, mixes with the venous blood of the vitelline vein. And I really want you to really grasp this idea, because in embryo, there is lots of mixing of blood. Okay, so embryo is never exposed to fully oxygenated blood, like 100% oxygen, never, because oxygen is toxic. Okay, so it's very toxic for the developing tissues. The maximum of oxygen that we keep in the embryo is about 94–96% oxygen. Okay, so which is a bit low, like compared to an adult, but it's fine. Okay, because it's proper for embryo. Okay, it's how we want it. So venous blood in the embryo is not the same venous blood as yours. It's still oxygenated. It has a normal amount of oxygen. It's just that it is a venous blood. So I hope this part was clear. Okay, so when we enter the sinus venosum, that's the first place where blood gets mixed. So we don't really expose the embryo to the proper 100% oxygen blood. Okay, so there's a little bit of mixing happens. So where does the blood flow after, right? So placenta, umbilical veins in the beginning, because don't forget, this is a 4th week. In the late placenta, in the late embryo, we only have one vein, right? We don't have 2 umbilical veins, we have 1. So this is 4th week embryo. That's why we have 2 umbilical veins. In the late embryo, there will only be 1 umbilical vein that is coming from the placenta. So where does the blood flow after? So this is the sinus venosus of the heart tube. From here, mixed blood goes to the primitive atrium. 1. Okay, there's only 1 atrium, primitive atrium. From the primitive atrium, the blood will go to the primitive ventricle, only 1. From the primitive ventricle, it will go to the bulbus cordis. Okay, there is little tiny part here which is known as the bulbus cordis. And from the bulbus cordis, the blood will flow to the truncus arteriosum. And this is the important part for us right now. Okay, so truncus arteriosum, the arterial end. Okay, so truncus arteriosum. The most distal part of the truncus arteriosum is known as the aortic sac. And this aortic sac is very important for making aortic arches. If you forgot, we're trying to understand what are these aortic arches and where do they come from. So again, heart tube of an embryo, the most distal end, sinus venosum, two horns, right, left sinus horn. Vessels that enter into these horns, umbilical veins, and for example, vitelline vein. So mixin appears here. Blood flows from the sinus venosum into the primitive atrium, from primitive atrium to the primitive ventricle, right? So everything's single. We only have one atrium, one ventricle. From ventricle to the bulbus cordis, from bulbus cordis to the truncus arteriosum, and the most distal end of the truncus arteriosum is known as the aortic sac. From the aortic sac, aortic arches start to appear. How many pharyngeal arches do we have?
5.
How many aortic arches are you expecting?
5.
5. And what will there be names? What will their names be?
First, second, third, fourth, and sixth.
And 6, precisely. The same as for the pharyngeal arches. Just you're going to call them aortic arches, right? So first you're going to see that first aortic arches, they're arches, right? So they're coming out like this. These are going to be first aortic arches coming from here. Then there would be second. Then there would be third, right? Then fourth. And then there would be 6 aortic arches. So total number is 5, but we call them 1, 2, 3, 4, 6 because the 5th one appears and disappears, right? So it is rudimentary. What makes these arches arches, right? So why do we call them an arch? Well, because it has a beginning and it has an end. The beginning of the aortic arches, this is the aortic sac. Where do all of the aortic arches drain into? They drain into a vessel that was formed by the vascular genesis, and these vessels are known as the dorsal aorta. So they come down like this here. This is one dorsal aorta left, and there's going to be another dorsal aorta here right. So I repeat, what makes an aortic arch an arch? It starts from the aortic sac and it drains into the dorsal aortas. So there is a right dorsal aorta and left. In the tail region, somewhere where the ass of the embryo will be, in the tail region, the two dorsal aortas will fuse together into one dorsal aorta. And unfortunately, I messed up here. The branch of a single dorsal aorta, the branch of it is umbilical artery. And where does umbilical artery go? It goes to the placenta. I know it's confusing, so we're gonna do it again, but on a different picture. Okay, so this is for you to read after. We're gonna do it on this picture. Okay, yours truly has been drawing this. So of course, for the purpose of the understanding of aortic arches, I'm not drawing here the sinus venosum or any of these parts, right? So I'm not drawing any of this region because it will only confuse us. It's part of the venous end of the heart tube. So what we have here, this is the truncus arteriosum, right? So this is the arterial end of the heart tube. And this is its distal end, which is the aortic sac, right? So this is the most superficial, the most superior part of the heart tube. The day is approximately day 25. So we start to see that the first aortic arch appears. On day 25, the first pair of the first aortic arches appears. It comes from the aortic sac like this. And what makes them an arch is that they drain into these vessels. How do we call these vessels? dorsal aortas. Okay, we're going to call them dorsal aortas. This is going to be the right dorsal aorta. This is the left dorsal aorta. These aortas have arisen via the vascular genesis. In the tail region, the two dorsal aortas fuse together, and they provide a single dorsal aorta that continues a bit down. But one of the branches of the single dorsal aorta is umbilical arteries, right? And these umbilical arteries, they are going to the placenta. So the kind of blood that they carry is technically venous, right? So because they are going to the placenta to drop the toxins, to remove the carbon dioxide, and etc. So branches of the single dorsal aorta are going to be your umbilical arteries. This little drawing here is trying to show you the position, how it is. So this is your heart tube. This tube right here is your gut tube. This is the vitelline duct, right? So this is the mid-gut region right here. This is the hindgut, this is the foregut, and this is the pharyngeal gut tube as an extension of the foregut a little bit, right? So you can see that the first aortic arches, do you see how they're positioning? They're kind of on the sides of the first aortic arches. So second will be here like this, right? Third would be here like this, like depending how your pharyngeal arches will continue. Behind your gut tube, we see the dorsal aortas, right? And somewhere, I made a mistake here, somewhere in the tail region, these two dorsal aortas will become one, right? And will continue down like this. This is day 25. This is day 26, the next day. So what do we see here? Still, we have your truncus arteriosum. The green thing is the aortic sac. These ones in violet color, right, purple, this is your first aortic arches. They have appeared on day 25. On day 26, in yellow, second aortic arches appear. So remember that your aortic arches, they appear consecutively, right? So they go in sequence, first and then second, and etc. So they don't appear all together like pharyngeal arches, for example, right? So day 26, this is when the second aortic arches appear. These arches, what makes them an arch, right, is that they come from the aortic sac and they drain into the two vessels in light green, which are the dorsal aortas. Two dorsal aortas in the tail region are fusing together into one single dorsal aorta and continue down. But on day 26, we also have additional vessels that are arising. They are called intersegmental arteries. Intersegmental arteries start to appear along the left and right dorsal aortas. Okay, so they are appearing on the right and left dorsal aortas. Day 27, 29. I point, you tell me. What is this?
Truncus
Truncus arteriosum. This one?
Aortic sac.
Aortic sac. This?
First aortic arch.
Aortic arches. This?
Second aortic arch.
This?
Third.
These ones?
Fourth.
They're arches because they start from the aortic sac and they drain into
dorsal
aortas. We have the right dorsal aorta here and we have the left dorsal aorta on the other side. Where do they fuse into one?
Placenta.
Where do they fuse into one? Where does right and left fuse into one?
Caudal end.
Tail region. So in the tail, in the caudal most end of the embryo, this is when the two dorsal aortas fuse into one single dorsal aorta. And what are the branches of the single dorsal aorta?
Umbilical artery.
And these umbilical arteries are going towards
Placenta.
And because they're going towards placenta, they are carrying venous blood. Well, not in your understanding venous, right? So embryo venous blood, it's still quite highly oxygenated, still has high oxygen, just it also has some carbon dioxide, urea, right? Because embryo does not technically pee properly, right? So it just has ureas that is taken to the placenta and etc. On day 27, third aortic arch appears. On day 29, fourth aortic arch appears, but something else simultaneously is happening. On day 27, first aortic arches stop being an arches. So they don't disappear, they don't degenerate, they just stop being an arches. So I repeat, what was making them arches in the first place? They started from the
Aortic sac.
and they drained into
Dorsal
aorta, the right and left on the two sides. So if they are no longer arches, that means that they no longer drain into the dorsal aorta. So what we're gonna see on day 27 is that the first aortic arches, they lose the connection to the dorsal aortas. If they lose the connection to the dorsal aorta, is there anything holding them down? Not really. So these two vessels start to ascend. They start to go up, okay? They will look like this. That's your first aortic arches. These first aortic arches after day 27, because they are ascending up, right? So they no longer drain into dorsal aorta, they ascend up, they become maxillary arteries. So after day 27, first aortic arches are becoming maxillary arteries. Now, second aortic arches do the same thing. Okay, so they do exactly the same thing. Just on day 29, they lose the connection to the dorsal aorta, so they no longer are arches, right? And they want to ascend. So they also want to ascend. And we see something like this here. So there's another second aortic arch that is going up like this. And this is going to be second aortic arch going up like this. And it's funny because the two second aortic arches, they are kind of splitting at the most distal end. They're kind of giving you two branches like this. Okay, so there are going to be two of them like this. The longer and bigger part, this is the hyoid artery. And this is the hyoid artery, right? So for the hyoid bone and stuff. And the smaller one, this is the stapedial artery for the stapes. Out of the two, one of them does not exist in you, and this is the stapedial artery, okay? You do have a hyoid artery, but you don't have stapedial artery. So later, stapedial artery should degenerate. And fun fact, if it doesn't degenerate, you will have auditory hallucinations because this artery will compress the auditory part of the vestibular cochlear nerve, and you're gonna have an auditory hallucination. You will hear voices. It's actually quite interesting case and very rare one, because if you hear voices, you know, schizophrenia, they will take you to the psych ward. But usually before they take you, they should do like an MRI, right? Or like a CT of your head to see. And sometimes the stapedial artery is visible. So all you have to do is like clip it, right? And remove it, and your auditory hallucinations will be gone. So first aortic arch will become maxillary, second aortic arches will become hyoid and stapedial arteries. Now, the little ones that we saw here, they were called intersegmental arteries, right? So, and we made them on day 26. So these were the intersegmental arteries. On day 27, 29, we're going to see that the first seven intersegmental arteries form a vessel which is going upwards. They form a common branch which is going upwards on two sides. The rest of the intersegmental, and we don't really know the exact number, just the rest of the intersegmental arteries will form a vessel which is going downwards. The seventh intersegmental is very important because it kind of supplies blood in both directions. The blood which is going up, right, and the blood which is going down. So seventh intersegmental has connection to both vessels, to the one which is going up and to the one which is going downwards. Okay, so remember this for now. Now, we have made the third, we have made the fourth. So what's interesting is this, that again, day 27, 29, this is dorsal aorta, and dorsal aorta specifically between the third and fourth aortic arches, so this region right here, this dorsal aorta starts to degenerate, it starts to disappear as well. As it is degenerating, as it is in the process of dying off, this part has a name. It is known as the carotid duct. So this question is usually a question for a 5. If they want to give you a 5, they will ask you this. So what is a carotid duct? The proper answer is carotid duct is a degenerating, it's in a process, it's degenerating dorsal aorta on both sides, which is located between third and fourth aortic arches. When carotid duct disappears, which arch is no longer an arch? So carotid duct is gone. Which arch is no longer an arch?
Second.
Second is here. So this is third. Okay, so the third aortic arch will not be an arch anymore. So I know this picture is confusing because this looks like as in one vessel, but it's not vessel. Okay, so I was trying to show that the first aortic arch is very thin, right? So it's like something is happening there. Second aortic arch right under it is also very thin. So this yellow one, this is the third aortic arch. And when carotid duct disappears, right? When carotid duct disappears, third aortic arch does not drain into dorsal aorta anymore because that part of the dorsal aorta is gone. That's it, right? So it's gone. So what's going to happen to the third aortic arch is the same thing. They will try to ascend as well. Why do we call it carotid duct? Because the third aortic arches are going to be carotid arteries, to be more specific, the common carotid arteries. So somewhere here, a bit lower, that's where we're going to have your carotid arteries. The third aortic arches are here. Now, as we know, common carotid arteries have two branches, internal and external. Which one arises first? So this is your third aortic arch here. Yeah, so third aortic arch is this one. Common carotid artery. Which branch would you make first? Internal or external, and why?
Internal.
Why internal?
Because it goes inside.
Exactly, because it's very important for the brain. Okay, so it's very important. So what is more important, face or brain? Well, it depends who you ask, but you know, brain is more important in my humble opinion. So internal carotid is the one which will be arising first, and it will just sprout out, it will just go very fastly towards the brain. And external carotid will slowly also start to appear, but much slower than the rest of the internal carotid. And the moment external carotid appears, this was first, right, aortic arch that became maxillary. And this was second aortic arch that became hyoid and stapedial. The moment external carotid artery appears, these two vessels will try to find the connection to it. They will, like in my drawing, it looks weird, but external carotid branch will be in the proximity of the first and second aortic arches. It will be very near them. So the first and second aortic arches are going to be attaching to the external carotid branch, and the rest of the aortic sac that was present here will degenerate, it will disappear. So this is the confusing part, right? Because we still had a bit of the aortic sac present here, from which the first and the second aortic arches appeared. So you're gonna see that that aortic sac will disappear. So you literally have vessels, right, that have no beginning, no end, like just little tiny vessels are just hanging somewhere in your body. So of course we need to reconnect to something, and these two vessels are actively searching for the external carotid branch of your third aortic arch. And internal continues to go up, just fully bypasses everything and just goes up, up, up. This part is a bit confusing, so yeah.
So aortic sac disappears when the third.
Not the whole thing, yeah. Do you see? So these are the third aortic arches, right?
Mm-hmm.
And this is the aortic sac. So this part of the aortic sac, this one, it will be overgrown by the third one. The third ones, they will, imagine they go up, yeah? So they go up. And as they go up, this region will become smaller and smaller and smaller. It will become kind of flattened out, kind of disappeared completely, because the third ones that are here like that, right? So that's third one, which is here, and this is another one here. They will do this because carotid duct is no longer holding them, so they do this. As they do this, this area of the carotid, it flattens this region of the aortic sac. So it will disappear, yes, under the weight of the third ones, because they will ascend. So under the weight of the third ones, it will disappear.
Thank you.
So next, as we can see, here are our third ones. The aortic sac between them is now flattened out. Do you see on this picture, right? So this is the green part of your aortic sac, and it's flattened out. Your first and second aortic arches are somewhere at the top over there, right? So they're looking for the connection to the external carotid artery, which is appearing somewhere here, right? Because third aortic arches are your common carotids. So somewhere up, you're going to see that the internal and external carotid arteries appear, and the maxillary or first aortic arch and hyoid or second aortic arch, they look for the connection to the external one. Now, here we have the fourth aortic arches appearing out of the aortic sac, and here we have the sixth aortic arches. And I really want you to see that something has happened to the aortic sac closer to the truncus arteriosum. Okay, so closer to the truncus arteriosum, aortic sac did something. And we saw it from this moment. Do you see there is a line here? So this is your one aortic sac. And there is a line here starting to appear on day 26. This line represents a septum, a future septum, and the septum is called aorticopulmonary septum. Take a look at your heart, your adult heart. It has two outflows, right? There is one circulation that is going for the aortic, and there is one which is going for the pulmonary, right? So they kind of go like this. One goes for the pulmonary. So it's a bit confusing, and it is part of the heart development, which we will discuss next lesson. But aortic sac is one. Inside of it, inside of the aortic sac, there is a little tiny spiral shaped septum starts to appear inside of it. And this septum slowly starts to divide your one aortic sac into two. So if I have to show, right, so this region of the aortic sac will be the future aortic region. And this part of the aortic sac is the future pulmonary. By the time 6 aortic arches appear, they do not come from the aortic part of the aortic sac. They come from the pulmonary part. They come from this area, and I try to draw it for you that this right 6 aortic arch, it kind of is behind your aortic sac, right? It comes from here. Does it make sense? Kind of. So one aortic sac is divided into two thanks to this particular septum. Again, it is not formed now, day 26, it's not there yet. I'm just trying to show you that in the future, you have to keep in mind constantly from day 26 that soon your one aortic sac will become two parts, two separate divisions, and it happens approximately now. So this is day 31. This is approximately day 31. We start to see that this proximal part of the aortic sac is divided into two. And the 6 aortic arches do not come from the aortic part of the aortic sac. They come from the pulmonary part. They come from here. This is why another name for the 6 aortic arches, they are also known as the pulmonary arches. Okay, they are pulmonary arches. They're waiting for the lungs to appear so they can supply them, because lungs will shortly appear here. So carotid duct is slowly degenerating, right? Third aortic arches are your common carotid, carotids are starting to ascend upwards. Fourth aortic arches, 6 ones, right? So this is the aortic sac right here. Very importantly, the nerves come down. The recurrent laryngeal nerves are coming down. And originally, recurrent laryngeal nerves are hooking around 6 aortic arches. Okay, so they come down and they hook around the 6 aortic arches, the 2 recurrent laryngeal nerves. Do you remember the vessels that we have formed by the first 7 intersegmental, right? So the first 7 intersegmental have formed a vessel which is going upwards. This vessel is the vertebral arteries. Okay, so the ones which are going up, they are called vertebral arteries. Once the vertebral arteries are formed, the first 6 intersegmental arteries degenerate. There's no need for them anymore because we have made the vertebral arteries. I know it's a bit confusing, but I repeat, we have made these vessels that are going upwards by union of the first 7 intersegmental arteries. Once we form this vessel, you don't need the rest of the 6, right? So you don't really need the first 6. So they start to degenerate. Do you remember we also made a vessel that was going downwards, right? So we also formed a vessel that was going downwards. We said that we do not know how many exactly intersegmental were used to make this vessel, but there is a vessel going downwards. The vessel that is going downwards, this is the internal thoracic. Okay, so this is going to be the internal thoracic arteries. When the first 6 intersegmental arteries disappear, the 7th intersegmental does not disappear, and quite on the contrary, it starts to grow and protrude outwards. On the 2 sides, the 7th intersegmental starts to protrude outwards. If this is the vertebral artery, and this is the internal thoracic artery, you can guess that the seventh intersegmental that is growing outwards on the two sides will become which vessel?
Brachial artery.
Again,
Brachial artery.
but the subclavian. Okay, so it's
Yes,
the subclavian, because specifically in the anatomy, you remember somewhere here, you see the subclavian artery go here and then vertebral up, and a little bit right next to it, the internal thoracic also goes down. There's like a particular region where you can see this. So the seventh intersegmental artery, right? So arteries on both sides will become subclavian. Just you have to remember the following. On the left side, this is the left side, the entire seventh intersegmental artery makes up the entire subclavian. So if I say this sentence, seventh intersegmental makes the entire subclavian artery. This statement is true, but only for the left side, only left side. On the right side, seventh intersegmental makes only a part of the subclavian. So some other arteries will help to make subclavian on the right side. So let's see. So far so good or no? Should I repeat something, come back? Okay, so we have 6 aortic arteries, pulmonary, right? 4th dorsal aortas right here, recurrent laryngeal. First 7 intersegmental have made a vertebral artery, right? So the rest made internal thoracic. 7th intersegmental grow outward and creating something which is going to be as the subclavian arteries. But it's true only on the left side, right? So it's going to be true only on the left side. On the right, it's going to be a part of it. So what happens next? What we see here, this is approximately day 34, 36. So how do we reach this part, day 34, 36? After day 31, right dorsal aorta, below the level of 6th aortic arch degenerates. I made a mistake here by drawing. So 7th intersegmental should be somewhere here. Okay, so somewhere here. I apologize for this. So this is 7th intersegmental. It should be at this level. It should be above the 6th aortic arch. Okay, so it should be a little bit above it. I don't know why I drew it like this, but I was young. So is this part clear? So 7th intersegmental should be somewhere here, above the 6th aortic arch. So I will show you which part disappears completely, right? So it's this entire region. All of this area, all of this right dorsal aorta degenerates. So below the level of the 6th aortic arch, the entire right dorsal aorta degenerates. If they ask you why, because the space here where the right dorsal aorta is located is going to be occupied by the inferior vena cava, because exactly at this moment we are forming inferior vena cava as well. And it takes a lot of space in the embryo. So right dorsal aorta starts to be compressed by the inferior vena cava, the one that we will make in the future, right? And the vessel just degenerates. Because this vessel degenerates, because right dorsal aorta degenerates, this entire region, so together with the 7th intersegmental, this entire region starts to ascend the same way as we saw it previously. Remember, if nothing holds you down, you start to ascend. So when this entire region starts to ascend, the 6th aortic arch on the right side loses the connection. It just simply detaches from the rest of the dorsal aorta. It just loses the connection. Do you see it here? Right? So 6th aortic arch no longer drains into the right dorsal aorta that we have a little bit of it here. It detaches and instead starts to supply the lung tissue. So this entire region starts to ascend upwards. And because it ascends upwards, it starts to shrink. Think about it this way. It was quite long, quite stretched out, right? Because of the right dorsal aorta pushing things down. When it disappears, this thing will like snap up, right? It will start to go upwards. And in the upward region, we have a little bit of space. It's not like we have lots of space. There's a little bit of space present there. Because of it, it starts to shrink. It shrinks all the way until this vessel, until it becomes this vessel. And this vessel right here is your subclavian. So we go back here. Where is subclavian on this picture? Starting from here and all the way until 7th intersegmental. Okay, so this is your subclavian right here on the right side. So a very important question is which vessels make up the subclavian on the right side? Distal part of the subclavian comes from the 7th intersegmental, right? So distally, it is intersegmental number 7. Middle portion of the subclavian, this blue color, as you remember, is the dorsal aorta, right? So this here is the right dorsal aorta between 4th and 6th aortic arches. So this is the middle portion of the subclavian. Proximal part of the subclavian, the little part of it, right? So this is going to be the 4th aortic arch, the one closer to the heart, right? So this is the 4th aortic arch. What about on the left side? The entire 7th intersegmental is your subclavian on the left side. So what are these? These are your 3rd aortic arches. As you remember, they're the common carotid. This is your aortic sac right here. A part of the aortic sac starts to protrude a bit more than the rest of it. Okay, there is no explanation. It just happens that it protrudes a bit more outwards. Probably this is your brachiocephalic trunk, of course, right? Because if this is the subclavian, this is the common carotid artery. So this part of the aortic sac that protrudes a bit more, of course, this will be your brachiocephalic trunk. How do you call this part of the aortic sac? Of course, this is the ascending aorta. So where does the ascending aorta come from? Aortic sac. This is the arch of the aorta. You have to be careful. Aortic arch, whenever you say aortic arch, they're thinking about these ones, yeah? They're thinking about 1st, 2nd, 4th, 6th aortic arches. So this is arch of the aorta, and it comes from the 4th aortic arch. A bit of it comes from the aortic sac, and a bit of it comes from the left dorsal aorta. The rest of the aorta, so your descending or thoracic or abdominal aorta, will come from the left dorsal aorta, right? So the rest of it is the left dorsal aorta. Now, what was recurrent laryngeal hooking around? Originally, it was hooking around the 6 aortic arches on both sides. But do you remember that on the right side, right, the 6th aortic arch lost the connection to the dorsal aorta. So recurrent laryngeal is going to jump up, right? Because now nothing is holding it here. So it's going to be jumping to the next thing that it can hook around, which is your 4th aortic arch. And we know that on the right side, the 4th aortic arch will become the proximal part of the subclavian. So this is why recurrent laryngeal nerve hooks around subclavian. If in the anatomy or in embryo they will ask you, that's the That's the explanation, right? So why recurrent laryngeal nerve hooks around subclavian? Because originally it was hooking around the 6 aortic arches or pulmonary arches, but 6 aortic arch on the right side has lost the connection to the dorsal aorta, right? So there is a gap right now here. So the next thing that the recurrent laryngeal can hook around, right? So which vessel is a bit up, is the 4th aortic arch. And we know that the 4th aortic arch on the right side is responsible for giving rise to the proximal part of the subclavian. Hence, recurrent laryngeal hooks around it. Now, what about on the left side? On the left side, right, so this is still your 6th aortic arch, which is pulmonary. Did it lose the connection to the left dorsal aorta? No, it still has it. And this connection between the 6th aortic arch on the left and the left dorsal aorta, so this connection is very important. It has a name, and it is called ductus arteriosus. Okay, so this part right here, this is ductus arteriosus, this connection between 6th aortic arch and left dorsal aorta. What is the purpose of the ductus arteriosus? Ductus arteriosus allows to bypass the lungs. Think about it. Do we need to send blood to the lungs for oxygenation? You are an embryo. Embryo is not breathing, it's not using the lungs. Where does oxygenation happen? In the placenta. So we need to make sure that if your heart sends the blood to the lungs, majority of it will bypass the lungs and go straight into the left dorsal aorta, from the left dorsal aorta into the umbilical artery, from the umbilical artery to the placenta, because this is where oxygenation will happen. So this ductus arteriosus here on the left side is a crucial vessel in embryo. If in embryo, for some reason, ductus arteriosus closes up too early, the embryo will die from suffocation. There's no way, right, to kind of get the blood going. Well, of course, I do not mean immediately. I mean that with some time, there will be less and less blood oxygen available and the embryo will die. So ductus arteriosus is very important for bypassing the lungs to make sure that blood does not stay in the lungs, right? So that it does not stay, that all of it goes to the left dorsal aorta, from left dorsal aorta to the placenta for oxygenation. You're born, you took your first breath, right? So your first breath as you were born, your lungs expanded with that first breath, and the smooth muscle tissue starts to secrete bradykinin. Bradykinin is responsible for contracting the smooth muscle in the ductus arteriosum. So ductus arteriosum closes up after you're born, because no need to bypass the lungs, you're born already, so of course it's time to use your lungs properly. So I repeat, how does your ductus arteriosum closes? With the help of the bradykinin. Who makes this bradykinin? Lungs, the smooth muscle tissue, smooth muscle cells in the lungs. Okay, so when you take a breath, your first breath, and then you continue breathing, your lungs keep expanding more and more bradykinin is made, more contracture of the ductus arteriosus happens, so it closes up and you're able to use your lungs properly. How do we call the remnant of the ductus arteriosum? It's called ligamentum arteriosum, and you have seen it in your anatomy practice. Okay, so this is a remnant of it. Okay. Are there any questions here? I will not continue more. I want you to take your time today and go over it again. I'm missing 20 minutes today, 20 minutes yesterday, so of course I'm adding it up to the next lesson, so don't worry. It's just that this is a lot for today. I really want you to go over it again, and tomorrow lesson we're going to start with it again. Okay, so you are going to tell me what is happening. I'm just going to ask you the questions, what is happening here, and you're going to tell me what's happening. So which day, what happened, what does the vessel do, and etc., etc., to kind of make sure that it stays in your head. Because what we did right now, we did like minimum 5 topics. Okay, so this is quite important. I forgot to mention here, last thing for today is that recurrent laryngeal on the right will hook around the proximal part of the subclavian, but on the left we say it hooks around the arch of the aorta, right? That's what we usually say in the anatomy. I had this topic with short Zoli when I was a second year, and I already knew embryology by that time, so I said that it doesn't hook around the arch of the aorta. Technically, it hooks around ligamentum arteriosum. And he said, no more questions. You have a 5, go home, because technically that's the proper way to answer. If you spread it out a little bit, I'm not sure if you have like possibility right now, like would you have open labs, or you don't have them anymore? Like, I don't know how it works anymore for you guys. But if you have an open lab and you have a chance to go and look at the arch of the aorta, right? So really try to spread this region right here, okay? Spread it apart. And if you will spread it apart, you will see that technically your recurrent laryngeal nerve, right? So the recurrent laryngeal, it doesn't really hook around the arch. It hooks around the ligamentum arteriosum. So on this picture, it's very spread out, right? So this drawing is very spread out. In real life, it's not that spread out. It's very close to each other. Your pulmonary arteries, right, and your arch of the aorta are very close to one another. So really dig in there, really like spread up, and you will see that technically the recurrent laryngeal hooks around this region specifically around the ligamentum arteriosum. So yeah, okay, we'll continue here tomorrow. Okay, so tomorrow it's going to be a bit, in my opinion, the most difficult out of them all, because we have to finish the venous system and we're going to do the heart. So really try to wake up for tomorrow's lesson. Okay, I will put the video in the group, and please remind me to highlight the topics that we did. I will send the screenshots, yes?
Thank you so much.
Thank you so much.
Thank you very much.
Thank you, thank you. Thank you.
Thank you.
Bye-bye, guys.
Bye-bye.
Thank you. Bye.